I want you to picture the health aisle at your local grocery store. You know the one I'm talking about.
Oh, yeah.
It is usually the brightest, most colorful aisle in the place, and honestly, it is screaming at
Your
you.
screaming promises, absolutely.
Exactly. Boost your immunity, defy aging, cellular defense.
Yeah.
And the word that is plastered on everything from pomegranate juice to face cream, the word we have been trained to worship, antioxidants.
It is arguably the most successful marketing term in the history of nutrition. It just has this incredible aura of benevolence around it.
And simplicity, that's the appeal, right? It sets up this perfect little comic book battle in our bodies. You have the villains free radicals, which I picture as these little biochemical terrorists blowing up our cells.
That makes a good image for it.
And then you have the heroes, antioxidants, the police force that sweeps in to save the day.
It is a very clean, very simple narrative. Easy to sell.
So, the logic follows. If radicals are the bad guys and antioxidants are the good guys, then it is just a numbers game. The more antioxidants I swallow, whether it's a pill, a powder, or a gummy, the safer I am.
That is the conventional wisdom. It is intuitive, it is comforting, and, well, it drives a multi-billion-dollar industry.
But
we are doing a deep dive today into the source material that underpins this whole concept, specifically Chapter 45, Free Radicals and Antioxidant Nutrients. And I have to tell you, if you actually read the biochemistry, that clean superhero narrative doesn't just get complicated.
no.
It falls apart completely.
And that is the hook for today. We aren't just going to talk about why blueberries are good for you. We are going to look at a terrifying reality hidden in the data.
Yeah.
We're going to talk about large-scale human trials where they took this more is better logic, gave people massive doses of good guy supplements like vitamin E and beta-carotene, and the result wasn't super health.
No, not at all. In some of these major trials, the result was increased mortality.
Increased mortality, people died sooner because they were trying to protect themselves.
Or they developed higher rates of cancer, specifically in the organs they were trying to save. It is a true biological paradox.
It sounds like a conspiracy theory, but it's right there in the hard science.
Yeah.
So our mission today is to unpack this paradox.
Right.
We need to dismantle the marketing and look at the actual mechanics. Why do our bodies rust? How do we naturally stop it? And why is taking a concentrated pill to override that system potentially like pouring gasoline on a fire?
And to answer that, we have to go really deep into the molecular machinery. We have a dense text here covering everything from DNA damage mechanisms to the specific enzymes your liver uses to hide heavy metals.
It is a lot, but it is fascinating. I am ready to get technical if you are. Let's start with the antagonist of the story. Section 1, the chemistry of chaos. What exactly is a free radical? Because I think most people just hear toxin and move on.
That is a fair assumption. But chemically, the definition is very precise, and it comes down to quantum mechanics, specifically electron spin. You remember high school chemistry.
Vaguely. I remember that electrons like to be in pairs. They have a buddy system.
That is the key. That's pretty much all you need to know. Electrons in atoms and molecules occupy these regions called orbitals. And thermodynamically, these orbitals are most stable when they're full.
Okay.
For most organic molecules, full means containing two electrons with opposite spins. They balance each other out.
Like a seesaw with two people of the same weight. It's balanced.
Exactly. Now, a free radical is any molecular species that has an unpaired electron. It has a vacancy. There's only one person on the seesaw.
And that makes it angry.
You could call it that.
Yeah.
It's chemically frustrated. It's incredibly unstable.
Yeah.
And this is where the danger lies in the time scale. I don't think people appreciate just how fast this damage happens.
Okay, what are we talking about here?
These species exist for a tiny, tiny fraction of time. We're talking about 10 to the minus 9 to 10 to the minus 12 seconds.
Hold on. 10 to the minus 12. That's a picosecond.
A trillionth of a second.
Okay, so before I can even blink, before a neuron can even fire in my brain to tell me I'm blinking, this thing has been born, lived its life, and died.
Well, not died. It has reacted. That is the scary part.
Yeah.
It forms, and it is so desperate to fill that empty electron slot that it will attack the very first thing it bumps into.
Yeah.
It doesn't travel through the cell looking for a specific target. It hits the nearest neighbor.
It's a molecular mugging, an instant smash and grab.
Instantaneously.
Yeah.
It steals an electron to stabilize itself. But think about the victim of that theft.
Right. If I am a stable protein, just doing my job, and you steal my electron to fix your problem,
Then you become the one with the unpaired electron.
so I become the radical.
Exactly. I become stable, but I have turned you into a monster. Now you have to steal from the next molecule.
And that molecule steals from the next.
It creates a self-perpetuating chain reaction of theft. One radical event doesn't just damage one molecule. It can propagate through a cell, damaging thousands of molecules in a domino effect until something stops it.
And the source says stopping it naturally is really hard, right? Because mathematically, what has to happen?
For the chain to break without an antioxidant involved, two radicals have to bump into each other.
Which, if they only exist for a trillionth of a second, and there aren't many of them in one place,
Is statistically incredibly rare. The concentration of radicals is low, and their lifespan is short. So they're much more likely to hit a stable molecule like a piece of your DNA, a protein, or a lipid, and damage that instead.
So it's just a path of destruction until they get lucky and run into another radical.
Pretty much, yeah.
Okay, so who are the usual suspects here? The source mentions ROS. What does that stand for?
That stands for reactive oxygen species. It is ironic, really. Oxygen is the breath of life. We die without it. But it is also the source of the danger.
The thing that gives us life also kills us. Classic.
The main characters we need to know are the superoxide radical, that's an O2 with a negative charge and an unpaired electron.
Superoxide, okay.
Then the perhydroxyl radical, which is O2H.
Okay.
And the nastiest one of the bunch, the hydroxyl radical, just an OH.
The hydroxyl radical, the source flagged that one as particularly devastating. Why that one?
It is the most reactive. It doesn't discriminate at all. It will attack the first nanometer of whatever it touches, whether that's the cell wall, an enzyme, or your genetic code. It's pure chaos. This cumulative damage caused by all these species is what we call oxidative damage.
Oxidative damage. It sounds so clinical, but what you're describing is just widespread vandalism on a microscopic scale.
That's a great way to put it.
So let's look at the casualties. This isn't just happening in empty space, it's happening to our biological infrastructure. The text highlights three main targets in Section 2: DNA, proteins, and lipids. Let's start with DNA because that sounds the most existential.
It is the most consequential in the long term, for sure. When a radical, especially a hydroxyl radical, interacts with the purine or pyrimidine bases in your DNA, the A, T, C, and G.
The letters in the book.
Exactly. It chemically modifies them. It changes the letters. It creates a typo in the genetic instruction manual.
And I'm guessing our bodies don't like typos in the manual.
Not at all. Now, our bodies are amazing. We have enzymes that run up and down the DNA looking for these typos and fixing them. It's called excision repair. But if that repair fails, or if the damage is overwhelming, that typo becomes permanent.
And then the cell divides.
It copies the typo faithfully. And every daughter cell after that has the typo.
So if this happens in, say, a lung cell.
If this happens in somatic cells, your regular body cells like longer skin, this is the initiation step of cancer. You have mutated the code that controls cell growth, and that's the first step down a very dangerous path.
But the source also mentions germline cells. That's a whole different level of problem.
Yes, that's the ovaries and testes. If the damage happens there, the stakes are different. The mutation isn't just in you.
It's in your kids.
It is passed down to your children. It leads to heritable mutations. So oxidative stress isn't just a personal health issue, it's a generational one.
That adds a whole new layer of weight to this. Okay, that's Battlefield 1. Battlefield 2 is proteins. And this part really surprised me. I usually think of protein damage as just structural, like the machine breaks and stops working.
And that can happen, for sure.
But the source connects this to autoimmune disease. How does that work?
This is a fascinating and somewhat terrifying mechanism.
Right.
Radicals can attack the amino acids that make up the protein chain. The text specifically mentions tyrosine residue.
Tyrosine.
When tyrosine gets hit by oxidative damage, it can be converted into something called DuP, or dihydroxyphenylalanine.
DuP, that sounds like a neurotransmitter, like dopamine.
It is related, but here it's a sign of damage. And DuPA can undergo further non-enzymatic reactions to create more radicals.
Yeah.
But the real problem is recognition. Your immune system has a catalog of self. It knows what your normal proteins look like.
It has the blueprints for every part of the machine.
Right. But when it sees this chemically modified oxidized protein, this protein with DOPA where tyrosine should be, it says, I don't recognize this. This is an alien.
So it attacks its own tissue.
It generates antibodies against the damaged protein. But here is the kicker. Because that damaged protein is still 99% similar to your healthy protein, those antibodies can start cross-reacting with normal healthy tissue.
Wow. So the wanted poster for the bad guy looks almost exactly like the good guy.
And the police start arresting everyone. The radical damage essentially tricks the immune system into launching a civil war.
That is wild. So rusting proteins can actually pull the trigger on things like lupus or rheumatoid arthritis.
It is a potential mechanism for initiating autoimmune disease, yes.
Yeah.
It links oxidative stress directly to immune dysfunction.
Incredible. Okay, battlefield 3. Lipids. Fats. The source calls this lipid peroxidation. And honestly, reading the description, this sounded the most gross. It's basically our cells going rancid.
That is exactly what it is. It's not just an analogy. It's the same chemical process.
Right.
If you leave a stick of butter out in the sun, or if you smell old vegetable oil that's gone off, that smell is lipid peroxidation.
And that's happening inside me.
It's happening in your cell membranes. Radicals target the unsaturated fatty acids, the ones with the double bonds.
And cell membranes are made of fatty acids. They're the walls of the city.
Correct. So the radicals steal electrons from the lipids in those walls, creating something called lipid peroxides. These are unstable, so they degrade into these highly reactive compounds called dialdehydes.
And dialdehydes are bad news.
They're like little toxic messengers. They are small enough that they can drift away from the membrane where they were born and go attack proteins or DNA elsewhere in the cell. They spread the chaos.
So the damage to the wall creates little smoke bombs that go and damage the buildings inside.
That's a good way to think of it. But here is the detective story part you liked. We can actually measure this happening in real time.
Yes. The breath test. This blew my mind. You're saying if my cells are rusting, you can literally smell it on my breath.
In an analytical sense, with the right equipment, yes. It depends on what kind of fat is being destroyed. The breakdown of N6 polyunsaturated fatty acids, like the ones in vegetable oils, produces a gas called pentane.
Pentane.
And the breakdown of N3 fatty acids, the kind in fish oil, produces ethane.
These are gases, right?
They're volatile hydrocarbons.
Yeah.
They dissolve in your blood, travel to the lungs, and you breathe them out.
So a scientist can put a mask on me, capture my exhale, run it through a machine, and say, whoa, your N6 fats are under heavy attack right now based on this pentane reading.
Exactly. It's a non-invasive window into cellular destruction. It's like a mechanic smelling the exhaust of a car to see if the engine is burning oil.
That is incredible. And the source mentioned some lab tests, too, for blood work.
Yes. We can use the FOX assay ferrous oxidation in xylanol orange.
Sounds like a sci-fi weapon from a video game.
It sounds cool, but it's just chemistry. It measures lipid peroxides by seeing how much they oxidize iron in an acidic solution. Or, more commonly, we can measure TBERs.
TBERs.
It stands for thiobarbituric acid reactive substances. It uses a chemical reaction that turns red and fluorescent if these dialdehydes are present. The redder the sample, the more rancid the lipids.
So you can literally see the rust in a test tube. It's amazing that we have these metrics.
It allows us to quantify the damage.
Okay, I want to connect this to the big killer, heart disease. This is section 3, the atherosclerosis connection.
This is a huge topic. And if we connect this to the bigger picture, it changes how we view cardiology.
I grew up thinking heart disease was a plumbing issue. You eat grease, the grease sticks to the pipe clogs. End of story.
Right, the simple plumbing model.
But the source material says that's wrong, or at least it's incomplete.
It is very incomplete. You can have high levels of LDL, the so-called bad cholesterol, circulating in your blood without developing plaques, as long as that LDL stays pristine.
Pristine, you mean? Clean.
Unoxidized. The problem starts when radicals attack the LDL particle itself. They modify the proteins and the lipids we just talked about.
So they rust the little cholesterol taxi.
Exactly. Now, normally, your liver manages cholesterol. It has LDL receptors on its surface that grab LDL out of the blood and process it.
It's like a recycling center for cholesterol.
Right. But the liver's receptors are very picky. They're like a lock and key. If the LDL is oxidized, if it's rusted and warped, the key doesn't fit the lock anymore. The liver does not recognize it.
So the liver rejects the trash. It just leaves it floating in the blood.
Exactly. It stays circulating.
Yeah.
And this is where the immune system makes a fatal mistake. There are other cells called macrophages, literally big eaters. They have a different kind of receptor called a scavenger receptor.
Scavenger receptor. That sounds less regulated, more like a garbage disposal.
It is completely unregulated. The liver stops taking in LDL when it has enough, has a feedback loop. The scavenger receptor on the macrophage does not have an off switch. It sees this weird oxidized LDL and thinks, I need to clean this up. So it eats it. And it eats another and another.
It just keeps eating.
It gorges itself.
Yeah.
Until it becomes what pathologists call a foam cell. It is literally so full of fatty droplets that under a microscope, it looks foamy.
A foam cell.
Wow. These cells eventually die from their gluttony. They are lipid-engorged macrophages.
And a dead fatty macrophage is basically plaque.
Yes, they infiltrate the wall of the blood vessel, specifically right under the endothelium, the inner lining. They accumulate, they die, they release all that fat and inflammatory signaling. That is the core of an atherosclerotic plaque.
So the root cause isn't just the cholesterol, it's the oxidation of the cholesterol that prevents the liver from clearing it, forcing the immune system to create a garbage dump in your artery walls.
Precisely. And this usually happens at sites where there is already a little bit of endothelial damage, maybe from high blood pressure, which lets those macrophages sneak in more easily. It is a perfect storm of damage and mismanagement.
This really changes how I think about diet and heart health. It's not just lower cholesterol, it's stop the cholesterol from rusting.
Which brings us to the obvious question. Where is all this rust coming from? Why are we full of radicals in the first place?
Right. Section 4. Where do radicals come from? We have external sources and internal sources. The external one is easy. Ionizing radiation, X-rays, UV light from the sun.
This is straight physics. High-energy radiation hits the water molecules in your body. We are mostly water, after all. It hits an H2O molecule with enough force to lease it to crack it open.
And cracking water open releases?
Hydroxyl radicals, immediately. That is why radiation causes radiation sickness and cancer. It is an instant flood of the most dangerous radical we know.
Okay, so avoid Chernobyl and wear sunscreen. Got it.
Yeah.
But the internal sources are the ones that really shook me, because they are happening right now while I'm sitting in this chair. The source calls it the leak in the power plant.
This is referring to your mitochondria. The power plants of your cells. They burn fuel using oxygen to create energy in the form of ATP. It's a controlled burn, passing electrons down a complex chain of enzymes.
But it's not 100% efficient. There are leaks.
No biological process is.
Yeah.
The source estimates that 3 to 5% of the oxygen you breathe doesn't get fully reduced to water like it's supposed to. It leaks out of the electron transport chain prematurely.
3 to 5%. That sounds small. Like, oh, I have a 5% error rate. That's not too bad.
It sounds small until you do the math. An adult consumes about 30 moles of oxygen a day.
Yeah.
If 5% of that becomes radicals, that is 1.5 moles of reactive oxygen species produced inside your body every single day.
I want to pause on that number. 1.5 moles. For the non-chemists listening, how many molecules is that?
A mole is Avogadro's number, which is 6 times 10 to the 23rd. So 1.5 moles is roughly 9 with 23 zeros after it.
900 sextillion molecules of poison every day.
It is a staggering amount of internal toxicity.
Just from breathing.
Just from being alive. It is the cost of doing business for an oxygen-based life form.
So living is technically fatal. Good to know. But there's another internal source that's even more surprising. We talked about the immune system fighting the plaque, but the immune system actually makes radicals on purpose.
Yes. This is called the respiratory burst. When a macrophage swallows a bacteria, it needs to kill it. How do you kill a bacteria inside a cell?
You poison it.
You bleach it, essentially.
Yeah.
The macrophage activates an enzyme called NADPH oxidase, also known as respiratory burst oxidase. It intentionally takes oxygen and converts it into superoxide. It shoots this radical blast at the bacteria to destroy its cell membrane.
So it manufactures the very thing we're trying to avoid and uses it as a weapon.
It's controlled chemical warfare. It uses a metabolic pathway called the pentose phosphate pathway to ramp up glucose use, which generates the NADPH it needs, and then it blasts the invader.
But wait, collateral damage. You can't set off a bomb in a crowded room without hitting the furniture.
Always. There's always collateral damage. And the source mentions a specific and very dangerous reaction here involving nitric oxide.
Nitric oxide, that's usually a good thing, right? It helps blood vessels relax.
Right. It's a critical signaling molecule.
Yeah.
But if nitric oxide meets that superoxide from the weapon blast, they combine to form peroxynitrite.
Peroxynitrite, that sounds evil.
It is. It's incredibly reactive, and it's unstable, so it decays into hydroxyl radicals, the worst of the worst.
Wow.
So when you are sick, when you have an infection, your markers of radical damage spike. Not just because the bacteria is doing something, but because you are doing it to kill the bacteria. That achy feeling you get during the flu, part of that is oxidative stress from your own immune response.
Unbelievable. And then there's the third internal source, transition metals. This is why we don't chew on iron bars.
Well, we need iron and copper for life. They're essential cofactors for enzymes. But free iron and free copper are dangerous.
What do they do?
They act as catalysts. They can interact with oxygen or hydrogen peroxide non-enzymatically to spawn radicals. This is often called Fenton chemistry.
Hmm.
If you have free iron floating around your blood, it acts like a radical factory, churning out hydroxyl radicals endlessly.
Which is a perfect segue to Section 5, the body's defense shield. Because if we are making 10 to the 23rd molecules of poison a day and we have iron floating around, why aren't we just a puddle of goo by age 5?
Because evolution is clever. We have a multi-layered defense system. The first strategy is simply containment. I call it hide the knives.
Hide the metals.
Exactly. Since free iron and copper cause trouble, the body never ever lets them float free. Iron is always bound to specific proteins. In the blood, it's transferrin. In storage in your cells, it's ferritin or hemosiderin.
And copper.
Copper is bound to a protein called ceruloplasmin. Others bind to metallothionein.
So it's like putting the gun in a safe. The metal is there, but it can't cause any trouble.
And if that safe breaks, if some metal ions do get free, the kidneys act as the backup. They are designed to filter out any free metal ions immediately. It's a very tight containment protocol to prevent accidental radical generation.
Okay, that's smart. But some radicals still get made. The leak happens, the immune burst happens, so we need a cleanup crew.
This is the enzymatic defense. We have specific enzymes evolved solely to disarm these bombs. The first line of defense is superoxide dismutase, or SOD.
I love that name. Dismutase. It sounds like it's calmly dismantling a bomb.
That's a good way to think of it. It takes superoxide, that initial leak product, and it catalyzes a reaction that converts it into hydrogen peroxide and oxygen.
Hydrogen peroxide, that's what you put on a cut. It's still toxic.
It is still toxic, but it's less reactive and less immediately dangerous than superoxide. So it downgrades the threat level.
So SOD passes the problem down the line.
And the next team comes in, catalase and other peroxidases. Their job is to take that hydrogen peroxide and break it down into water and oxygen. Completely harmless. So it's a two-step process to neutralize the threat.
Nice. And the text mentions glutathione peroxidase too. What's its special job?
That one is crucial for the lipids we talked about. It specializes in reducing those nasty lipid peroxides in your cell membranes back to harmless hydroxy fatty acids. It stops the rancidity in its tracks.
So it's the fat-soluble cleanup crew.
Exactly. But here is the key dietary connection. Glutathione peroxidase is a selenium-dependent enzyme.
Selenium, that's one of those minerals on the back of the multivitamin bottle I usually ignore.
You shouldn't. Without selenium, this enzyme cannot function. It is a core component. It's the fuel for the fire extinguisher. That's why selenium is an essential nutrient. If you are selenium deficient, your lipid defense system shuts down.
That's a really clear connection. Okay, so we have metal safes and we have enzyme squads. But sometimes a radical slips through, especially into the cell membranes where the enzymes can't always reach easily. That's where the famous vitamins come in. Section 6, the vitamin relay race.
I like that image because it really is a relay. It's about passing the buck from a dangerous place to a safe place.
Let's start with vitamin E. The source calls it tocopherol.
Yes. Vitamin E is lipid soluble.
So it lives in the fat.
It lives inside the cell membrane. It is right there in the trenches where the lipid peroxidation happens. When a radical comes along and attacks a fatty acid, vitamin G steps in and says, "Take me instead."
It jumps on the grenade.
Yes. It donates its electron to quench the radical chain reaction, saving the lipid. But now we have a physics problem. Vitamin E has lost an electron. It has become a radical itself, the tocopheroxyl radical.
Wait, so we just traded one radical for another. How does that help? Why isn't the vitamin E radical just as dangerous as the one it replaced?
That is the million-dollar question.
Okay.
And the difference is its molecular structure. The source points out figure 45-3. The unpaired electron in the vitamin E radical can be delocalized.
Delocalize. Unpack that for us. What does that mean?
Imagine you're holding a burning hot coal. If you hold it in one spot on your hand, it burns through instantly. But if you could juggle it really, really fast or spread the heat out across your entire body instantly, it wouldn't burn you as much.
Okay, I can picture that.
The vitamin E molecule has a ring structure called a chrominal ring. When it gets that unpaired electron, it doesn't just hold it in one place, it spreads the instability across 3 different positions in that ring. It shares the burden.
So the radical nature is diluted, in a way.
It makes it relatively stable. It doesn't instantly attack its neighbor. It waits. It's less reactive.
It holds the hot potato, but it can't hold it forever. Something has to take it away.
No, it needs to be recycled. This is where vitamin C or ascorbate comes in. Vitamin C is water-soluble. It floats in the fluid outside the membrane.
In the aqueous phase, as they say.
Right. It comes up to the surface of the membrane, meets the vitamin E radical holding that hot potato and says, give me that electron burden.
The handoff, the baton pass in the relay.
Exactly. Vitamin C donates an electron, reducing the tocopheroxyl radical back to active, healthy vitamin E. Now, vitamin E is ready to go back to work.
But now vitamin C is the radical. We've just moved the problem again.
True. Vitamin C becomes the monodehydroascorbate radical. But vitamin C is in the water. It is easily flushed out by the kidneys or recycled by other enzymes in the cytoplasm, like NADH-dependent reductases. It is much safer to have a radical in your urine than in the structural walls of your cells.
So it's a bucket brigade. Radicals hit the fat, vitamin E catches it, passes it to vitamin C at the surface. Vitamin C gets washed out or recycled by the main system.
Precisely. And this interplay explains why you need both. One protects the fat, the other resets the protector. If you only have one, the system clogs up.
This sounds perfect. It's an elegant, beautiful system. We understand the damage, we understand the defense, so logic dictates. If I take a massive pill with 1,000% of my daily vitamin E and C, I am supercharging this relay race. I should be invincible.
And that is the logic that sold 1 billion pills.
But here's where it gets really interesting.
And here we arrive at section 7, the antioxidant paradox. The moment where that simple, beautiful logic fails spectacularly.
The text is explicit about this. It says antioxidants can act as pro-oxidants. That sounds like a betrayal, a firefighter who is also an arsonist.
It is chemically inevitable under the wrong conditions. Let's look at vitamin C again. We just praised it. But the text shows in table 45-1 that if you have high concentrations of vitamin C in the presence of free copper ions,
and very metal we're supposed to keep locked up,
vitamin C can react with that copper, C2 place material, to produce superoxide and hydroxyl radicals.
So instead of quenching radicals, it starts making them.
In high doses and the presence of a catalyst like copper, yes. If you flood the system with vitamin C, you might overwhelm the metal binding capacity or just increase the statistical probability of this reaction.
So more isn't always better.
Now, in the body, your kidneys are really good at this. They usually pee out excess vitamin C once your plasma concentration hits roughly 30 micromoles per liter, so it's hard to reach those toxic levels from food. But it highlights the chemical duality.
The real tragedy, though, is the beta-carotene story. This is the one that really shocked the medical world.
The beta-carotene failure. This is a profound lesson in scientific humility.
Take us back to the 90s. What was the hypothesis? Why did everyone think beta-carotene was a miracle nutrient?
In the 90s, we looked at epidemiological data. Big population studies. We saw that people who ate lots of carrots, sweet potatoes, and leafy greens, people with high levels of beta-carotene in their blood, had significantly lower rates of lung cancer, even among smokers.
Correlation. Carrots equal less cancer. It seems straightforward.
So logically, scientists thought, it's the beta-carotene. It's a powerful antioxidant. Let's extract it, purify it, put it in a pill, and give it to the people at highest risk, smokers and asbestos workers. We thought we were going to prevent lung cancer.
It makes perfect sense. Smokers have incredibly high oxidative stress from the smoke. Give them the shield.
They ran 2 major intervention trials, the ATBC trial in Finland and the carrot trial in the U.S. Thousands and thousands of participants, and they had to stop both trials early.
Why? Not because they were working so well, I'm guessing?
Because the people taking the beta-carotene supplements were dying. In the ATBC trial, they had an 18% increase in lung cancer incidence and an 8% increase in overall mortality compared to the placebo group.
That is horrifying. You give someone a pill to save them and you accelerate their disease? Why? How does an antioxidant cause cancer?
The source explains it clearly, and it comes down to partial pressure of oxygen. Beta-carotene is a radical-trapping antioxidant, yes, but it functions best at low partial pressure of oxygen.
Like in your muscle tissue or deep inside the body?
Exactly. But where are the lungs, especially a smoker's lungs?
They are full of air, high oxygen pressure.
The highest oxygen tension in the body. And at high oxygen pressure, and especially in the high concentrations you get from a supplement, beta-carotene flips its allegiance. It becomes an autocatalytic pro-oxidant.
These flips.
It reacts with the oxygen to initiate radical damage to lipids and proteins rather than stopping it.
Yeah.
In that specific environment, it becomes fuel for the fire.
That is terrifying. Context matters. Chemistry changes based on the environment.
And then there is the vitamin E problem.
Yeah.
It wasn't as dramatic, but meta-analyses of multiple trials showed increased mortality with high-dose vitamin E supplements too.
Why? Is it the same pro-oxidant effect, or is it the relay race again?
It is the broken relay. Imagine you flood your cell membranes with synthetic vitamin E from a supplement. It does its job. It catches the radicals. It becomes the vitamin E radical.
Okay.
But what if you don't have enough vitamin C to recycle all of it? What if the supplement dose of E is way out of proportion to your dietary C?
You have a traffic jam of used up, radicalized vitamin E.
And remember, we said the vitamin E radical is relatively stable. That sounds good, but it means it survives long enough to drift. It acts like a ferry.
A ferry. A ferry for what?
It can penetrate deeper into the LDL particle. It carries the radical burden from the surface, where the water-soluble vitamin C could have reached it, deep into the core of the cholesterol molecule.
Oh, no. It hides the fire where the fire extinguisher can't reach.
Exactly. It becomes a Trojan horse, ferrying the oxidation into the heart of the LDL, initiating lipid peroxidation from the inside out. So by taking just the vitamin E supplement, without the balance of the whole system, you are effectively injecting the damage deeper into the building.
This brings us to the final twist, Section 8. Because there's another, even more profound reason why stopping all free radicals might be a bad idea. We might actually need them.
This is the most profound shift in thinking in this entire field. We used to think radicals were just waste products, metabolic trash. But biology doesn't really do waste. It uses everything.
Radicals are signals.
They are distress signals.
Yeah.
Specifically for a process called apoptosis.
Apoptosis. That's programmed cell death. Cell suicide for the greater good of the organism.
Exactly. When a cell has suffered too much DNA damage, maybe it's on the path to becoming cancerous, it needs to be eliminated, it needs to die. How does it know to die?
I have no idea.
The accumulation of radicals inside the cell acts as the trigger. It's the fire alarm. A rising tide of oxidative stress is one of the key signals that tells the cell your damage is beyond repair. It's time to self-destruct.
So the radicals are screaming, "We are broken. Shut us down before we hurt anyone else."
And the cellular machinery hears that signal and initiates apoptosis, killing the cell before it can become a tumor. It's a fundamental anti-cancer mechanism.
Okay, I see where this is going. What happens if you flood that damaged cell with high-dose antioxidants from a supplement?
You quench the radicals. You turn off the alarm.
You mute the signal.
You mute the signal. The cell is still damaged. It still has the DNA mutations. The fire is still burning. But the die signal never goes off. So the damaged cell survives.
And it divides, and it passes on its damaged DNA, and now you have cancer.
That is the hypothesis for why these antioxidant trials often show increased cancer rates. By protecting the cell from oxidative stress, you're also protecting the damaged cells from being eliminated by the body's own quality control. You are helping the enemy survive and replicate.
That is heavy. It completely flips the script. We aren't just rusting cars that need a coat of paint. We are complex cities with signaling networks, and if you cut the phone lines, disasters happen.
It illustrates the danger of reductionist thinking. Radical bad, vitamin good is too simple for a system that has been evolving for 1 billion years. The body is a balance of generation and quenching, signaling and silence.
So what does this all mean? Let's bring this home for the listener who is staring at their medicine cabinet right now, full of brightly colored bottles. Outro time. What is the takeaway?
The takeaway is that biology is smarter than marketing. Free radicals are inevitable and destructive, yes, but they are also necessary signals. We have elaborate, evolved systems to handle them. Enzymes that require specific minerals, sequestration proteins, a vitamin relay race that requires balance. When we intervene with massive, unphysiological doses of isolated nutrients, we disrupt that balance in ways we can't always predict.
So it seems like the best antioxidant isn't a pill, it's a functioning, healthy metabolism.
It is eating the whole food, where the vitamin E comes packaged with the vitamin C and the polyphenols and 1,000 other compounds in the right ratios, not a concentrated chemical isolate from a lab.
The rust analogy is too simple.
It's a delicate signaling network, and we're tinkering with it without reading the manual.
I'm thinking about that leak in the mitochondria again.
Yes.
1.5 moles of radicals a day. We are walking nuclear reactors. It's a miracle we survive at all.
It is. The control systems are breathtakingly effective. And it makes you wonder, if the body works so hard to pee out excess vitamin C and to lock up every last ion of free iron, are we just a little bit arrogant to think we can boost this system with a pill?
Maybe the goal shouldn't be to stop the rust, but to trust the machine that was designed to handle it.
I think that's the wisest conclusion. Support the machine, don't try to override it.
Trust the machine. I like that. I'm going to go eat an actual carrot instead of a beta-carotene capsule.
Very wise choice.
Thanks for diving deep with us today. Check your sources, check your breath for pentane, and we'll see you on the next one.